כשחשבנו על התוכנית, חשבנו על 3 רבדים. אחד מהם רובד שהוא הרובד של אנשים שהתחנכו עם עמירם, עבדו עם עמירם, כאשר היינו סטודנטים או פוסט-דוקים, ורובד שני הוא הרובד של תלמידות ותלמידים של עמירם, ורובד שלישי הוא הרובד של אנשים שממשיכים את התחום ופורצים דרכים חדשות עכשיו. אני מבקש להזמין לבמה את פרופסור יוסף שלזינגר, יוסי, שבכינויו אצלי תמיד, אל תגיד יוסף, כן, שלז, שהוא מדען ידוע מאוד בעולם בתחום שהוא עכשיו יחבר אותו לחקר המוח, ואני מאוד מודה ליוסי על זה שהגיע לכאן מניו הבן ומצא את הדרך.
מקליפורניה, דרך קליפורניה.
מקליפורניה?
דרך קליפורניה.
וואו. תודה רבה.
Well, it's nice to be here, but it's sad circumstances. I have been a friend of Amiram for 55 years, and I'll tell more about our friendship at the end of the lectures. I would emphasize initially aspect of the science and what led to what, and then talk also about the long interaction that we had since we met when each of us were about 20 years old. So this is a very long-term relationship. So what I'm going to tell you today is several stories. I'm going to tell you the main topic of my work, which is receptor tyrosine kinase as targets for cancer and other therapeutics. I'm going to describe a unique mechanism of ALK, it's a receptor tyrosine kinase by monomeric and dimeric ligands. I will show that the ligand of these molecules, once you figure that out, okay. אני צריך להסתכל על המיקרופון, okay. Shows a role in metabolism and pain, and this is how it linked to the nervous system. And finally, localized ligand-induced ALK activation in neuronal tissue, which I think has to do with unique mechanism how it works. And I'm going to say at the end also 55 years of friendship with Amiram. So this is the family of molecules that I've been studying for 45 years. I started with very similar to what Amiram was, a spectroscopist, biophysicist. We did PhD in the same lab. Even the postdoctoral training that we did was done, I did at Cornell on measuring diffusion coefficient using fluorescence, and Amiram did it at Yale. But I realized that I became interested in membrane receptors, and I also realized that we are facing a real molecular revolution of molecular biology. And I decided to marry those two and basically focus on one aspect of molecule. And all of you know about insulin receptor and about other receptors. So in the late 70s, it became clear that this is a very important family of molecules. So I decided to focus most of our research on that. And I did everything possible, discovering them, isolating them. Most of the research I did initially on EGF receptor and others, establishing the mechanisms, studying how they work and all that. So I'm not going to elaborate on that because I want to show you first the potential of this knowledge for human therapy. And also elements of the mechanism which were very crucial. And this shows you the mechanism that we proposed, which is a very simple mechanism, that namely these receptors are largely monomers, and in order to activate and transduce signal across the membrane, they have to go dimerization. And so we studied everything, but then it became clear that there are a lot of lesions which are driven by mutation in these receptors and how they signal. And since the early 80s, I became involved in starting companies about it. So just to give you some general description of what is the social impact of this research, I'm going to show you the drug discovery activity which I've been involved, and then focus on a specific story, which is kind of unique and new, some of it unpublished, which is going to link to neurosciences. So this has been my drug discovery activity in the last 45 years. So I founded 6 biotech companies. The first one was Sugen. This was the first biotech company to develop small molecule kinase inhibitors for cancer treatment, and this led to 2 FDA-approved drugs. The second company which I founded was about every 10 years. It's called Plexicon. I became also very heavily involved in structural analysis. I was educated and trained as crystallographer, more recently as cryo-EM. This is structure-guided drug kinase inhibitor, 2 FDA-approved drugs. Then I realized that I missed an opportunity by not working on therapeutic antibodies, because we were the first to generate therapeutic antibodies, but then I missed this opportunity. And in 2008, I founded a company which is based on therapeutic antibodies. It eventually merged with another company called Celldex, and we have now a phase 2 clinical trial, a drug in the disease of urticaria. So this shows you some of these drugs that have been developed. Sugen, these are the 2 drugs that have been developed by Sugen, Xalori and Sutent. They are large market drugs, and some of them is already expired, as the patents are already expired. And the second company is Plexicon, and these are the 2 drugs that have been developed. At a certain stage, I realized that in order for the companies to be involved in a way that I have some say, since the second company, I'm the chairman of the board of all the companies I founded. So I'm an amateur entrepreneur, although 85 of my efforts are into breaking new grounds in science. So this is the 4th company I founded, called Inozyme. At a certain stage, I realized that some people have better ideas than me, and this is another idea that I have been involved with, which is also linked to my science. This is an enzyme replacement therapy for rare fatal disease of aberrant calcification, and there's a drug already in phase 2 trial. Opna Bio is a very exciting new company which could be a major breakthrough in oncology. It's based on the discovery of Doug Hanahan, which also is a potential link between neurosciences. He has shown that FMRP, other one known as Fragile X mental retardation protein, is a major driver of cancer cell immune suppression in tumors. So FMRP is a master regulator that controls protein translation and mRNA stability of hundreds of genes. Tumors with high FMRP levels are immune deserts, largely devoid of T cells, while FMRP deficient tumors are inflamed. So we are working on drugs that block this molecule, and the idea that this will be a universal immuno-oncology drug. And last but not least, this is a company I founded very recently. We're still financing it, some of it from my own. This is an endocrine hormone, which we were working for many years. We are using structure to do structure-guided potent FGF21 variants for the treatment of pancreatitis, alcohol toxicity, as well as treatment of subset of cancer. So this just shows you, for those of you who are not scientists, if you spend a lot of time in doing basic science, which this is what I'm primarily doing, and you find new targets, this has quite immediate significations for drug discovery. Not immediate, it's a 40 years effort. So I found myself doing that. So now I'm going to show you a new story which has to do with this receptor tyrosine kinase, which are rather interesting, called ALK and LTK. So I'm going to show you everything from how they work, what is their function, and how they are linked to metabolic processes which are regulated in the hypothalamus and in pain in dorsal root ganglia. So it's going to be a link where mechanistically I'm going to show you the details. So this is this receptor tyrosine kinase called ALK. It has 2 members, ALK and LTK. ALK has the following hallmarks here: large extracellular domain, a single transmembrane domain. It was initially discovered as a major oncogene of multiple cancer, lung cancer, neuroblastoma, thyroid cancer. But it was clear a receptor, but no one knew what the ligand. Therefore, we called it for 20 years orphan receptors. So since it was a receptor that drove cancer, a variety of drugs were developed, and some of the drugs are one of those which I mentioned to you, and this is successful in using for treatment of certain patients. And so this is, for example, how these molecules are mutated in different cancers. You can see here that there are ALK fusion proteins which are causing a variety of cancers here. There's also mutations, point mutation, and this is a list of the different cancer, melanoma, neuroblastoma, certain lung cancer and all that. And one of the drugs that we have developed is successful. But so it's molecule, it's interesting, it's important, but then how does it really work? What is the ligand? So for more than 20 years... לא שומעים אותי? אז אני אצעק.
אוקיי, זה תשים בכיס.
אוקיי, זה יותר טוב ככה. אוקיי, ככה אני לא צריך לי... Okay, so as mentioned, for more than 20 years, ALK was designated an orphan receptor that functions as a driver of several cancers, but no one knows how it really works. And there was several false reports providing insight, but they were refuted, so we did not know what it is. About several years ago, a hint came from a company for a ligand for LTK, which we immediately took advantage, and we were able to purify and discover two ligands of this receptor, which we call augmenters. It has multiple names. One of them is a dimeric molecule. We mapped the cysteine. We did all the biochemistry. Augmenter is either a covalent disulfide or non-covalent dimer. Augmenter beta is a monomeric molecule, and it was already surprising how a dimeric ligand and a monomeric ligand can activate a receptor, considering that we have established this general rule that receptor dimerization is essential. So we solved this problem, and there are really only about 10 papers about this topic. This is what I like, because then I can really move and offer totally open new foundation. So this is basically showing that if you take a portion of the receptor, this region here, this can activate the receptor at all without even having a dimer. So this was a surprise, how a fragment of the receptor can activate it, which is certainly a monomer. And this shows you this experiment, that when you take a wild type augmenter or the derivative, which is molecule which is conserved, it can activate both ALK and LTK. So a monomeric ligand or a fragment of the molecule are as potent. This was surprising, against the dogma that I sort of was involved. So we were basically thinking what's the explanation, and this is this publication, but I think we came to the true answer, because this is what we proposed in that paper, and that is, taken together, this experiment showed that a deletion of the N-terminal variable region minimally affects the activity of augmenter alpha toward LTK and ALK stimulation in culture cells. Therefore, reduced dimerization might be compensated by high local concentration of augmenter AD bound to ALK in the cell membrane and by potential ligand-induced receptor-receptor interactions. And so I wouldn't show it if I wouldn't prove that this is the mechanism. And so how we really challenge it is by doing structural analysis. So we spent a lot of time studying the mechanism of action of these molecules at atomic resolution. We solved several structures, established a mechanism, and so far we have quite a few of them. And this also helped in developing drugs. So now I'm going to show you how the structure of this molecule. This is a little elaborate, so I'm going to make it a rather simple slide in order to be able to visualize it for you. So this is the region, which is a ligand-binding region that we solved. It has this 3-helix fold, glycine-rich region, TNF-like repeat, and EGF1. So this is the ligand-binding region that the structure had been determined. And this shows you this unique region here, which is basically what we call hexagonal array of polyglycine helices, and it's sort of spectacular structurally and visually. So this is really this region here. Look how it really looks up in terms of the atomic interactions. It's really beautiful. We didn't find it in any other protein, and we have no clue why it's that. but this is something which is sort of unique. So this is how the unoccupied receptor looks like. So now we're going to show how ALK and LTK are activated by their physiological ligands, which is full-size augmentor, this dimeric molecule which I showed you earlier. And so this shows you the structure. It's complicated, it has multicolor. I'll spend a lot of time to it because I know that this is new. So this is the membrane here, and these are the two ligand molecules. And actually, some of the element of the structure were not visualized. So this is how the dimer looks like, but in the cryo-EM, we were not able to visualize this region, but this is what, it is there, but it's probably too mobile. And here we have two receptors, and we have basically colored different regions in different color to show clarity. So what I'm going to show you, you can see that this molecule actually lies on the membrane, and I'm going to show you the multiple interactions which really result for this receptor activation. It's rather comprehensive, extremely sort of detailed, but I think it contains a lot of insight. So this is basically, again, what it is, and I have marking all the different regions here which are causing interactions which are required for dimerization. So the first site is basically a primary ligand receptor site, which is here. There is a secondary, more small receptor site of the ligand. There are receptor-receptor interactions. And moreover, uniquely, which has never been seen in any receptor-target kinase, the ligand itself interacts with the membrane. So I'm going to now open these modules and show you individual. This is rather new. This was just published some times ago. So this is basically, now I'm opening the ligand receptor, and in red, you can see the interface in the ligand that binds to the interface in the receptor. So this is the primary ligand region here. When it's closed, it's looking like that. It has a very large buried surface area, about 1,000 angstrom, square angstrom. So this is basically, so there's three, we published 2 papers in Nature, and there was a third one from another lab. So there is total consensus about the primary ligand binding region, which is the one I showed you so far. And basically, the cryo-EM is much more detailed analysis, and these are 2 crystal structure. So now I'm going to show you the other interactions. So this is an open book again, where I'm peeling it off, and I'm using red to show you the region in the ligand and the region in the receptor. So you can really see how a receptor and ligand form multiple interactions which eventually stabilize a formation of an activated state. And this is very complicated. It's very difficult to digest it, but that's what it is. Sometimes nature is not simple, although the interactions themselves are simple. So this is another, and here you see another interface, and these are the receptor-receptor interaction. I've already described in red, the peeling it off and showing how it interacts. So you can start to build up 3-dimensional interaction by this and this is a much smaller interface. And finally, this is the last interface here, which shows you another thing which is dramatic, and that is that the ligand binds, and then there is this structural chains which lead to the receptor. So this is this multiple interaction. It's totally different than other receptor targets in kinase. I did not include any of that in my lecture because this is more than enough for people who are an expert, but this is kind of unique. So we have to ask you a question, why is it so unique? So this just shows you again the summary. The molecule lies on the membrane, and one of the reasons why it interacts with the membrane is because the ligand itself has these multiple ligands that interact on the membrane. So this is clearly a unique mechanism. To most of you, it doesn't make any sense anyway, but I want to tell you that we think that it has to do with the function of this molecule. And this is what I'm going to show you in the next few slides. But before that, I still want to give a visual manifestation of how a monomeric ligand and a dimeric ligand can activate, which was basically contradicting or sort of related to a dogma that I have been pushing forward for the last 40 years. And it shows you here how that can happen. And basically you can see here, this is a dimeric ligand, but this is a monomeric ligand. And here, because the multiple low-affinity interaction and strong interaction of the membrane, because of dimensionality effects, which is sort of more complicated way of describing it, both a monomeric ligand and a dimeric ligand can act the receptor. So the question I want to ask now is why is ALK activated by a unique mechanism of activation that is so different from the mechanism of activation of most RTKs by their ligand? You have to take my word. I've been working on many of them and all that. This is really different. Why is it different? Well, it's different because you have to study the biology. ALK is uniquely activated in different neuronal tissues. And we did not know what the function is until we generated mice which are deficient in the ligand and in the receptor. Another lab generated ALK, and we generated it. This is basically a study we published last year, and we showed that the ligand acts and the receptor act primarily in the hypothalamus. And this shows you here a hypothalamic pathway for augmented alpha controlled by weight loss. And we showed clearly that in the knockout mice are thin, and this is entirely regulated by event that took place in the hypothalamus. So remember now, this is an oncogenic receptor. So a pathway, a neuronal pathway, was co-opted to induce multiple cancer. We can discuss that too. And since it's published, I'll summarize you the take-home messages of this paper. And that's basically it. Augmento alpha is expressed in the hypothalamus in what we call AGRP neurons of acute nucleus, ARC. Starvation causes strong augmento alpha expression in AGRP neurons. So when you starve, the expression of this molecule is induced. Moreover, augmento mice are thin. Augmento mice fed with high-fat diet also lose weight. A similar phenotype was detected for the receptor. So it's clearly a regulator of metabolism. But that's not all. Thinness of augmented mice is associated primarily with enhanced metabolic parameters and locomotion. These mice are the main reason for the thinness because they're very active. Athletic mice somehow. And moreover, localized augmento alpha induce activation of PVN neurons in the hypothalamus. And this is something which is unique here and different. So we have to focus on what is known in certain regions in the hypothalamus which regulate metabolism. And that slide shows you a scheme of a leptin-dependent mechanism where you have the so-called AGRP neurons, which are produced, and then projections of this neuron act on regions where express melanocortin receptors. So this neuron regulates hunger, and this neuron regulates satiety. And the balance between these molecules. Well, it turned out that ALK is expressed here, and the ligand is expressed here. So it's very different than other receptor tyrosine, because the ligand behaves a little like a neurotransmitter. It's released locally. It doesn't really, it's not a circulated hormone. It's released locally, and then it really acts. So this is basically what this part of the story is, and we are studying with detail, is that augmento alpha is locally released by projections of AGRP neurons with a paraventricular nucleus, and this is shown simply here. So we are really studying how is it regulated, why is it regulated, so there's no evidence for circulation of the ligand. It is just released, and then it probably glides on the membrane. So this provides to me a lot of interesting secrets that I would like to evaluate. While we thought that we start to understand, came out at second paper. And this paper shows you that the neuronal tyrosine kinase, which is another name for augmento alpha. There are only about 20 papers about this topic, but this is another name. So this molecule seems to be involved in the control of pain. So what was shown in that paper, that augmento alpha, the ligand that we're talking, is expressed in TRP1 DRG neurons. This is neurons which are induced by caspacin mechanisms, and augmento alpha induce phosphorylation of ALK in spinal dorsal horn, and they showed that drugs that act on ALK reduce significantly pain. So here we have another mechanism. And this is one of the drugs that they use is the inhibitor we developed. So when these patients are treated for the cancer, they have less pain. So it was thought that maybe because the burden of the cancer could also be that it acts on the target. So this was sort of very interesting. So we had the killer experiment. We have the knockout mice, so we can see what happens to pain. So we trained our lab to measure this mechanism, and I can show you here that augmented KO mice and ALK inhibitors reverse thermal hyperglycemia pain. And we are checking it an additional mechanism of pain, and here you see the experiment. These are all unpublished data. Effects of. So this shows you, and we educated ourselves. I got a collaborator, which is an expert on pain. You can see clearly that the sensitivity to pain of augmento alpha is about 3-fold reduced. And same thing, we repeated the experiment with drugs. So this is very interesting. We do have also evidence that augmento alpha is induced in the dorsal root ganglia. Okay, you can see very nice expression. You can see that there is low level of ALK and a high level of LTK, which is the other receptor. So there could be, so this is another local mechanism of ligand and a receptor that control pain. So we are very busy now trying to understand how it works, and I'll just show you a few slides until I wrap up. And this is just another example, what I showed you. Again, a local action of augmenter with a receptor. This is why this probably mechanism is so unique. So the structural analysis revealed the mechanism of ligand-induced activation of ALK and LTK by this region. The question is, what is the function of the N-terminal region of ALK? This region here. So we know how it's activated, but then this extra region. And this is this region here. So this is how it's activated. We know everything here, but we don't know. So we decided, and this is, I'm going to wrap up the lecture, just give you a little points about the future. And that is, so we decided to see what happens to this region when you stain cells, neuronal and non-neuronal. So we made fluorescent variants. So we are going back to fluorescence, like what me and Amiram were doing many years ago. And so this is the probe of the N-terminal region of the molecule, which we made with halothane, fluorescent, purified it, and we started to stain cells. And we saw very quickly with cells which express this are not neuronal cell, which express ALK, that it binds to heparin sulfate proteoglycan. If you use wild-type CHO cells, it's strong binding, and CHO cells which are deficient in heparin, no binding. If you take the wild type, you can block them with heparin. So clearly, it binds probably to heparin-rich regions in neuronal cells. And it's some kind of domain region. So I will leave you at there. This shows you how it sort of interacts with PC12 cells, which are induced by NGF receptor. By NGF stimulation, you see that there is domains. And here is another experiment with PC12 cells showing you the same thing. And then we also isolated dorsal root ganglia, which show the same thing. So we are now looking into a mechanism how this process is regulated to ion channel and to the TRP receptor. And actually I met in California David Julius, which we discussed the possibility of linking this molecule because neurotrophic factors and apparently ALK are involved in similar pathways. So it really, once you follow your science, you can go to totally different territories, and that's what I'm trying to do. So cell surface localization of the N-terminal region of ALK in heparin sulfate proteoglycans region on cell membranes and in discrete regions of neuronal projection. So summary, I told you a little about receptor tyrosine kinase and their targets in cancer. The cryo-EM analysis, which was relative new education of my lab, showing you these multiple interactions. The knockout mice tell us, go to the neuronal system, go to hypothalamus, go to dorsal root ganglia, and then the mechanism of ALK activation is consistent with local ligand-induced receptor activation in neuronal tissue, which opens a totally new area which we are going to explore. Now, a few words about 55 years of friendship with Amiram Grindelwald. I am going to insist on Amiram. I first met Amiram when both of us were about 22 years old, and so we, in the chemistry department in the university, we initially did some, we stayed in different places, we initially did some lab together, in physics lab and chemistry lab, and developed sort of friendship. And then at a certain stage, we decided to move together and share a dormitory. So in the third year, after the Six-Day War, we both living in a dormitory in the Givat Ram. And then when we started to do master degree, we continued and spent. So we're 3 years together, staying together. When we decided to look for postdoc for graduate school, we went to the Weizmann Institute with Avram Napastek to really hunt for potential. And we were quite impressed that all the big guys there wanted to talk to us. So we both decided to go and do PhD with Yitzhak Steinberg, whose widow is here. Before she was married. She was married at the same month that we came, and I was also married to my first wife at the same month. So this was in August. What year was it? Anyway, so Amiram and I were very much interested in quantitative science. I sort of became a little qualitative by becoming molecular biologist, but I think I did sufficiently well with it. But we continuously communicated. There was some kind of hidden competition between us. We were both very competitive, but there was a strong affection. So, as you know, Amiram was a MP Betzanchanim, K'tzin Khan. I was the MP of Khan in Golani. And there was one event after the Yom Kippur War where Amiram was stationed in Mazrat Bijan. And my unit left Mazrat Bijan. So I heard that so I looked for him, and we were really hugging each other 20 kilometers from Damascus. So we shared a lot of military experiences, and this is typical aspect of our generation. We also came from rather beaten up families for many reasons. There was tragic element in his life. There was significant tragic element in my life. But we were both excessively optimistic and really ambitious. And in many ways, his ambitious fueled mine and vice versa. We really cared for each other, and there was never issue of jealousy, but because we just liked each other. Things even really got improved in a significant way when we got older. We became much more mellow, and there was all those years of friendship between us. I followed his illness, and I tried to do my best, sometimes in terms of advice, but Amiram wouldn't listen. But I really miss him. 55 years of friendship is unique, and we are sort of examples of children of Holocaust survivors that try to survive and thrive, did well in our profession, and develop along the life very strong bonds. Thank you very much.
יוסי, המון תודה על הרצאה מרתקת וסיום מרגש. לא יודע אם זה המקום, אבל במלחמת יום הכיפורים, אני בגלל סיבה אחרת לגמרי, הגעתי לחרמון, ואז אמרו לי, אל תלך לכיוון ההוא, יש שם מ"פ הנדסה שיורה בכל מה שמתקרב. זה לא היה עמירם. בכל מקרה, המון תודה, זה מרגש. כן, יש משהו בעובדה שאמרת על דור של משפחות ניצולות שואה מתקרב יותר לתחום של מה טראומה עושה בדור שני ובדור שלישי, אבל זה נושא אחר לגמרי, והוא כבד ואנחנו לא נמלטים ממנו בארץ הזה, ולא נמלט כל כך מהר. במיוחד לאור העובדה שאפיגנטיקה מראה שזה יותר מורכב ממה שאנחנו חשבנו פעם. אם יש שאלה מדעית כלשהי, זה הזמן לנושא של יוסי, ואם לא, אני מניח שאנשים יכולים לשאול אותך אחר כך, ואני באמת תאסיר תודה, יוסי, שהגעת. יש שאלה, סליחה. דפנה. פרופסור דפנה אטלס, תקבל גם מיקרופון, אם כי אני מכיר אותך, את יכולה גם לי מיקרופון. אוקיי, לא. אז רגע, אתה פה.
אני אחזור.
תשאיר את זה, תחזור.
אני דפנה אטלס מאוניברסיטה העברית, אבל בילינו יחד את
Another 50 years friend.
בילינו ביחד את ה-PhD, זה היה נחמד, איתך ועם עמירם. ראיתי בסלייד שלך שדיברת על ציסטאין 66, על הקשר SS בון, נכון? עכשיו, אם אתה נגיד שובר את הקשר הזה, מה קורה לפעילות?
לא משתנה. הדומיין שלא קושר בכלל את האזור הזה, מספיק פעיל. אחת הסיבות שאני עובד על זה ששום דבר לא ידוע, זה יש 2-3 מעבדות. אנחנו מנסים לראות אם ההורמון הזה הוא פרו-הורמון, ואם יש איזשהו אקטיב, הוא דרך אגב מתבטא בסקרטורי גרנולס, והוא עובר סקריטר על ידי membrane potentials. There's a lot of interesting things which are going on here. And there could be a possibility that it is just a pro-hormone and we have a fragment. But basically, this is really new. This is why I like it. Well, I cannot retire nor die until this is understood.
I hope it's not understood soon.
Okay. Okay.
יוסי, המון תודות.